There are a few clarifications and additions that I'd like to make to the previous video that you watched about vesicular transport. It provided a good overview despite being a little weird and not knowing whether they were talking to students or talking to teachers. But in any event, the information presented was both accurate and comprehensive, so I thought it was a good video for you all to watch. But as I said, a few clarifications. The first point that I'd like to make is that if we're beginning with a donor compartment, the location and orientation of the things, proteins specifically, that are in that donor compartment are going to be maintained throughout vesicular transport. In the top donor compartment, you can see soluble proteins represented by red dots and membrane proteins represented by little lollipop type things. The membrane proteins are oriented such that the lollipop head is in the cytoplasm. We'll call the outside of that compartment the cytoplasm. And the stick portion of the lollipop is in the membrane with the end of the stick in the lumen of that donor compartment. Of course, the soluble proteins are in the lumen of that compartment. As a vesicle buds off, that orientation of both membrane-bound proteins and soluble proteins is maintained such that what had been in the donor compartment oriented toward the cytoplasm remains oriented toward the cytoplasm in the budded vesicle. What had been oriented toward the donor compartment lumen remains oriented toward the lumen of the vesicle. That orientation is maintained after fusion, such that in the target compartment, what had been oriented toward the cytoplasm remains oriented toward the cytoplasm. What had been oriented toward the lumen of the donor compartment is now oriented toward the lumen of the target compartment. Anything that was in the lumen of the donor compartment, those soluble proteins, are going to be delivered to the lumen of the target compartment. This is our secretory pathway, and we're going to focus on moving from the endoplasmic reticulum through that secretory pathway through the various stacks of the Golgi and leaving the transface of the Golgi to move toward the plasma membrane. That is what we call anterograde transport through the secretory pathway. One thing that we didn't mention when we spoke about the secretory pathway in the past is that it is true that things will move or proteins will be delivered to the plasma membrane, to the cell surface. But what we didn't say was that it doesn't have to be by default. If it is by default, that unregulated delivery to the cell surface is considered constitutive secretion. I've noted it here, and it's also highlighted on the slide. Constitutive secretion is movement from the trans-Golgi via a vesicle by default to the cell surface. It's also possible, however, that what leaves the transface of the Golgi might remain in a secretory vesicle that's stored behind, if you will, the plasma membrane until a specific signal is received. This signal might be electrical excitation, so what would be contained in the secretory vesicle would be a neurotransmitter, or there might be a hormone like insulin in that secretory vesicle that you don't want to release unless there is information coming from outside of the cell that blood glucose concentrations are high. In that case, the vesicle will receive a signal and it will then move toward the plasma membrane, and the contents of that vesicle will be released into the extracellular space upon fusion. One thing that is missing from this diagram is showing that there is such a thing as retrograde transport, backward transport. Obviously, that might be from the cell surface through endocytosis, and anything endocytosed would end up in an endosome and ultimately potentially in a lysosome. Retrograde transport does also occur between the stacks of the Golgi, moving from the trans face through the medial to the cis, and in fact, retrograde transport does occur from the cis face of the Golgi backward toward the endoplasmic reticulum. As the video said, there are multiple types of coat proteins that are responsible for facilitating the formation of vesicles. Those three flavors of coat proteins are shown in that legend. There is clathrin, COP1, and COP2. COP stands for coatomer protein. And again, we have coatomer protein 1 and coatomer protein 2. I'd like to focus on coatomer protein 2 and the mechanism by which coatomer protein 2 facilitates the formation of vesicles at the endoplasmic reticulum for transport of things to the cis face of the Golgi. As the video said, we're going to need to recruit these COP proteins, these coatomer proteins, to, in this case, the ER membrane. That recruitment is going to occur because of a monomeric G protein called SAR1. What the video left out is that SAR1 contains an amphipathic helix that is hidden when SAR1 is in the GDP bound form. When SAR1 GDP approaches the ER donor membrane, it will encounter a guanine nucleotide exchange factor for SAR1, the SAR1 GEF, and that exchange factor will cause the release of GDP and the binding of GTP. When SAR1 is in its GTP bound state, the amphipathic helix is now exposed, and the hydrophobic side of that amphipathic helix associates with the outer face of the ER membrane. Of course, SAR1 will ultimately hydrolyze that GTP, and when it does, it is going to resume the GDP bound conformation, which will preclude its association with the ER membrane. We'll see how this is involved in vesicular transport in just a little bit. Once embedded in the cytoplasmic face of the ER membrane, SAR1 then recruits the adapter proteins, specifically Sec24 and Sec23. These adapter proteins then are responsible for recruiting the coatomer proteins. Those coatomer proteins will assemble on the adapter proteins, and due to the shape of the coatomer proteins, will deform the membrane, creating a bud. Here we'll add another adapter protein and coatomer protein, so you might imagine that a vesicle would begin to, the neck of that vesicle would begin to get smaller and smaller. We'll add another here, and then we'll add the dynamin that ultimately will twist around the neck of that vesicle to cause the vesicle to bud off. Once the vesicle has bud off the ER membrane, that is when hydrolysis of GTP by SAR will occur. That hydrolysis prevents, as I said, SAR1 from associating with the membrane, and now the vesicle membrane. And because SAR1 cannot associate with the vesicle membrane, the adapter proteins cannot associate with the vesicle membrane, nor can the coatomer proteins associate with the vesicle membrane. So all of those coat proteins and adapter proteins and recruitment proteins fall off or disassemble, leaving what we call a naked vesicle. The naked vesicle, when a vesicle becomes naked, it will expose the proteins that are going to be required for docking onto a target compartment. The previous video spoke at length about this, so I won't go into detail, but recall that Rab proteins are responsible for tethering and docking, and snare proteins on the vesicle as well as on the target membrane are going to be responsible for vesicular fusion to the target membrane. I want to go into a little more detail about how the snare proteins facilitate vesicle fusion with the target membrane. And it's simply because the V snare on the vesicle and the T snare on the target membrane twist very tightly with one another. Their twisting forces water that is between the vesicle and the target membrane out of that space, bringing the vesicle membrane and the target membrane in close opposition to one another. On the left side, you can see how this might happen so that you would form initially a stalk between the vesicle and the target membrane, and eventually on the right-hand side, complete fusion. So what would have been in the vesicle is now delivered to the lumen of the target compartment. These were the clarifications that I hoped to make to the previous video. Now you know all you need to know about vesicular transport.